Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today Dr. James Brown, who is the co-founder, president, and CEO of DURECT. DURECT is pioneering a therapeutic strategy by leveraging the potential of epigenetic modulation to alter the trajectory of severe and life-threatening conditions like acute organ injury and cancer. The company is specifically focused on the development of an epigenetic modulator called larsucosterol, or DUR928, for the treatment of alcohol-associated hepatitis, or AH. The therapeutic has shown promising results in human studies, including a Phase IIb trial in AH patients. Dr. Brown co-founded DURECT in 1998 and has served as president, CEO, and a director since 1998 as well. Dr. Brown has worked in various leadership positions, including as vice president of Biopharmaceutical and Implant research and development at ALZA Corporation from 1995 to 1998, and various leadership roles at Syntex, which is now Roche. Dr. Brown holds a Doctor of Veterinary Medicine from the University of California, Davis, where he also conducted postgraduate work in pharmacology and toxicology. To learn more about Dr. Brown's work and that he's leading at DURECT, let's turn to him now. Welcome to the show, Dr. Brown.
Oh, thank you, Ayesha, and please call me Jim. Thank you so much. I really appreciate that wonderful introduction.
Thank you so much, Jim. I'd like to first start off by taking a step back and really learning more about your background in the industry. You've had such an illustrious career in the field, and so I'd like to learn more about that and also what led you to co-found DURECT.
Yes. Well, I started life as a practicing veterinarian. I still have my license and enjoy doing that. But what led me into the pharmaceutical industry, I'd worked before I went to veterinary school. As an undergraduate, I'd worked for a diagnostic company called SYVA, SYVA, which is a joint venture between Syntex and Varian, developing various diagnostic tests. And that gave me my first insight into industry. And then when I'd graduated, I was in private practice for a while as a veterinarian. And what really led me to get back into industry was I had a client who, she was an older client who had one son who had died in the Vietnam War, and her husband had died, and she had only her old cat as her family, and she was in a rent control department that wouldn't allow new pets, and so it became extremely important to keep that cat healthy and alive. And I realized as I was working with her and just my experience in practice that I was treating the animals, but in reality, I was helping the people, right? And that what I looked at and what I aspired to do was to maybe reach out and try and help more people in a broader way. And so that's why I got into developing new drugs, new pharmaceutical agents.
Wonderful. Thank you so much for sharing that story. And I really love that, how helping pets and how that helped you transition also to helping humans directly. So you were helping indirectly and directly.
Yeah, right.
And so I'd like to learn more about DURECT's missions and goals. And I know that the company is focused on addressing unmet needs in indications specifically like alcohol-associated hepatitis. And so, so what are the missions and goals of DURECT? And I guess, secondly, what are the unmet medical needs in the indications that you're pursuing?
Our focus is to develop epigenetic therapies to reach diseases for which there are no good therapies today. And unfortunately, there are a lot of these type of diseases, especially in the acute side, which is where we're starting. We all know about the circumstances around sepsis or stroke or acute pancreatitis or acute kidney injury. And all of these circumstances, there really aren't good therapeutics out there. There are things that the physicians can do for you that, they can provide supportive care and help you through a circumstance, but up to a large extent, the body itself has to be able to overcome and protect itself and come out the other side. Well, it's the same circumstance for alcohol-associated hepatitis, which is the lead indication that we are pursuing for larsucosterol. But when we look at all these diseases, the first four or so I mentioned, what makes it challenging for those diseases is using stroke as an example. If I were to have a twin brother that had a stroke, and we both had the same stroke at the same time, and he was to get to a hospital 12 hours ahead of me and just get supportive care, he would have a better outcome than me. And then if we were both in a clinical trial, even though we had the same lesion at the same time, he'd have that advantage. And so then if one were randomized to active and one were randomized to the placebo group, you have different, you have more noise, more in a clinical trial. And that's been really the ultimate challenge for conducting trials in sepsis and stroke and a number of these other conditions, is that biological variability just brought on by timing of these acute circumstances. Well, with alcohol-associated hepatitis, or AH, the circumstances are slightly different than that. One isn't on this 12-hour clock to be able to intervene. Unfortunately, there really isn't much that can be done for these patients. And to put it in perspective, there are in the United States about 160,000 hospitalizations per year for AH. We recently conducted a trial that had over 230 patients from the United States in it, and our average age was 44 years old. And these people die at a rate of 30% over the next 90 days. This is a very real circumstance. 20% of the population are young women in their 20s and 30s who don't have cirrhosis, but there is more binge drinking now. There's more fatty liver. You put those two things together, and you have a greater potential for this. And so this is a highly lethal disease for which there is no therapy. But the reason we selected it is because there's a huge unmet need. We're talking almost as many people, 30 to 40,000 people dying every year in the United States. That's almost as many people as die from breast cancer or from automobile accidents, and with no therapy out there today. And yet we have the time to be able to talk to these patients, to interact with their families, and to get them to consent to be in the trial, because unfortunately they're on a circumstance where they're on this kind of slow train wreck to death, and so we have time to be able to try and dose them and see if we can help.
That's very enlightening in a way where I think a lot of us don't really think about how grave the repercussions are of this disease and how prevalent it is. So
it is. Yeah, it's so much more prevalent. And it's interesting, Ayesha, until I started working on a potential therapy for it, I didn't know much about it either. And yet now I'm sitting on a train going between New York and Boston, or on an airplane, or at a wedding reception, talk to people and find out a brother, a sister, someone had it and died from it. It's definitely out there.
I just think it's not a condition that maybe is at the forefront. Maybe a lot of people don't speak about it. There's stigma and all of those issues associated with
that.
It.
Definitely is the case. Yeah, with alcohol use disorder, generally speaking, we don't talk about it much. And even people die from this disease. They typically die at the end of the day from kidney damage, because it's like having a heart attack from the liver. The liver fails to the point where it's shedding toxins and kills the kidneys. And so sometimes the death certificate won't say alcohol-associated hepatitis. It'll say renal disease or something, because they kind of want to spare the family that circumstance. So as a society, we definitely don't approach it that way and aren't talking about it as much as we should. But it's a very real, horrible circumstance out there.
Absolutely. And so that makes DURECT's work so ever important. And I'd like to learn more about the company's approach to epigenetic modulation and how your lead asset addresses the underlying cause or the underlying root of AH.
Yeah, it definitely does. The way larsucosterol works is it inhibits a group of enzymes that are called DNA methyltransferases. The acronym is DNMT. And what they do is these molecules put methyl groups onto DNA. And the way DNA is expressed, it's read along a line. They call it a gene cassette back from the old days of tapes, where the methyl group prevents the cassette from being expressed, and the gene cassette. And so what larsucosterol does is it goes in and it changes the methylation. And typically in patients with AH and in a number of disease states, you get hypermethylation. So you get a lot of genes changed. Some are turned on, some are turned off by virtue of the process because there's a cascade of events. We know that larsucosterol influences about 70 major nuclear pathways that influences things like inflammation, like cell survival, improving the mitochondrial membrane, stabilizing that, and improving things like autophagy, which is kind of like the garbage collection within the cell, cleaning up the cell's debris. And so there are a lot of things that are in disarray during states of hypermethylation in circumstances like with AH that are put right and brought back more towards normalcy and allow the cells to survive.
And so I know that the compound is in human trials, which is very exciting.
Right.
And so could you tell us a bit more about the trial results and the data you have so far and what stage of development it's at currently?
Yeah, it's currently, we're on the cusp of starting a phase 3 trial, would be the last phase of development. We have done a lot of safety work with the molecule. We've dosed over 500 people in various diseases and circumstances to understand the safety of the drug. And the drug is a very nice safety profile. But most importantly, were the results that we have from our Phase IIb trial, which we call AFFIRM. And AFFIRM is a trial of 307 patients that all have serious AH. That means they have alcohol-associated hepatitis with a MELD score between 21 to 30. And what does that mean? Now, MELD score is used to stage a patient for liver transplant. So it kind of gives you a sense of how sick the patient is by virtue of their liver disease. And if you have a MELD of 21, that means you would have about a 20% chance of dying in the next 90 days from your disease. But it's a fairly steep slope. If you go up to the MELD of 30, you have about a 60% chance of dying. And so that's the realm that we're working in with these patients. We ended up in our trial having a MELD range in the 24 to 25 range. Our mortality in the U.S., historically, we expect the mortality, 90-day mortality for patients in that range to be about 30%. And in the United States, it was about 28%, very close to what we expected. And the U.S. data were nothing short of really remarkable. We had 232 patients out of 307 enrolled in the United States, where we have very relatively uniform healthcare provision here. And we tested 2 different doses of the drug. So we dosed either placebo or dose 30 milligram dose or a 90 milligram dose of larsucosterol. And we saw about 57 to 58% reduction in mortality in each of those 2 doses, which were highly statistically significant for the US data. Where we ran into issues was outside the United States. This was conducted during the pandemic. And so in order to keep enrolling patients at that pace, we expanded it to healthcare systems outside the U.S. where the diagnosis and the treatment and the patient journeys are just different for AH, as you would expect, unfortunately. So that led to a dampening of the signal. We still ended up with about a 40% reduction in mortality, but it wasn't quite statistically significant for one dose and about a 35% for another that was farther out. And so what we're doing right now is planning in the planning stages of doing a phase 3 trial that would be conducted just here in the United States. And that would be our objective, would be to get that trial started by the end of the year. And if we do, we'll be able to we think we should have data by the end of 2026. And so that's our objective. And it's interesting, the way this drug is dosed, because it's an epigenetic regulator, it's dosed on day 1. And if they're still in the hospital 3 days later, it's dosed again. And then we just follow the patients for 90 days. So it's not something you give every 6 hours or every 12 hours or even once a week. You just give it once. And if you think about it, that makes sense, right? You're changing.
Yeah, once you change the methylation patterns. Yeah.
Right. Yeah. I mean, for people who are non-scientists, I use the analogy of a computer where I talk about, because the nucleus of the cell, a very small amount of what's in the nucleus of the cell is DNA. less than 5% of what's in the nucleus of the cell. The rest is the epigenome, which controls the expression. It's the same with the computers we're using. A very small amount of the computing capacity within the computers we're using is the chips and the hardware. It's the software that allows for the expression of it, right? And so what we're doing, if you consider the epigenome to be the software, you don't have to have the geek squad out to your house to redo your computer every 6 hours, hopefully, right? If you got a problem, you have the person come out and she or he can fix it, and you're good for the next month or two, hopefully. And so that's what we're looking at.
That's a great analogy, that the epigenetic regulation is akin to software of a computer, and you don't need to keep updating or keep running that. And and so what is the durability like for an epigenetic modulator like this?
It's a great question. You would think it would be just the life of the proteins that were expressed, right, in relatively short, which maybe a week or two or less, right? But the durability, the effect is layered. So if you can imagine, when you're in these circumstances, you've got acute hepatitis that's going to kill you. Like the best analogy I can think of is like a heart attack of the liver, right? So the liver is dying. And you give them a drug that can change the methylation state of these cells, stabilize the mitochondrial membrane, reduce inflammation, increase and reduce apoptosis, and increase autophagy, and all these kind of things that are influencing the capacity of cells to survive. Then the consequences of that are much longer, because then those cells live, they produce less toxin, so the renal cells, are less damaged, and then they can repair more readily. We can give the drug. We've given it orally, topically, by injection, and in this case, we give it intravenously because we want to get to various organs. Because when these patients die, they don't just die of liver disease; they die of multi-organ damage, and so we want to protect against that. And we've shown dramatic multi-organ protection from this molecule. There is a test that we did in rodents with Tylenol, with acetaminophen, and if you overdose with acetaminophen, you can die, right? I can tell you as a veterinarian, if you give your cat 325 milligram Tylenol, you'll kill your cat because they don't have the enzymes to be able to handle it. But what it does is it damages the liver, it damages the kidneys, it damages the lungs, it damages the GI lining. A whole bunch of different things go on. And when we give our molecule to rodents that have been exposed to high doses of acetaminophen, if you don't give the drug, the vast majority of these animals, 90% die. If you give the drug, 90% live. And if you look at the organs, those livers and lungs and kidneys look normal. It's amazing in its capacity to protect. So it's a very long-winded answer. I apologize, but it's multi-layered. Yeah. It's a multi-layered circumstance.
Right, and so it's targeting the protein or the gene. I missed the name of it. Is what specific gene does it target, or is it a host of genes?
There's three enzymes called DNA methyltransferase.
Right.
There's subtypes, 3 of them. There's it doesn't matter, but there's 3 of them in mammals, and their role is to put methyl groups onto DNA. And so what larsucosterol does is it inhibits those dramatically. And we know, for example, in AH patients, you can look at the literature and you can see those enzymes are really high. So we know there's hypermethylation going on. We know those enzymes are up. And then so we give larsucosterol, it brings that down, and it changes the hypermethylation state. And then that in turn influences a lot of different genes, because in some years ago, there was a shampoo commercial where they had a person in a box, and she told 2 friends and they told 2 friends. Do you remember that commercial? Maybe it's probably too long ago. Never mind.
But I get it. It's kind of like, like the telephone kind of effect, or
it's that kind of thing, right? Where you get this amplification that 2 friends tell 2 friends, and then they tell 4 friends, and 8 and then 16. It's that kind of thing, like
word of mouth,
division of bacteria. Yeah, right. And so when you look at methylation and you change methylation, that will cause some genes to be turned off, and some genes to be turned on. And if you look at the ones that are turned off and turned on, they in turn influence other genes, which in turn influence other genes. Yeah. And so you go down that cascade, and it ends up being more than 1,000 different genes are turned on and turned off. Okay. So the biology is tremendously complex. And the reason we're able to develop this drug from our perspective right now is because it's a naturally occurring molecule. that we have been part of discovering and using in that way. We know that it's made in association with the mitochondria under mitochondrial stress, travels to the nucleus, and then has its effect. And so by virtue of doing that, we can have all these beneficial effects, and we've really seen very little toxicity. In fact, in these patients in this trial, we actually saw about 20% fewer, they call treatment emergent adverse events in both of the dosing arms where the drug was as compared to a placebo, because there were just simply fewer issues in these patients in the ICU. So they actually had a safer journey in the hospital. So you have not a benefit risk ratio for the FDA to be concerned about, but rather a benefit circumstance. And I think that plus the unmet need is what led the FDA to grant us breakthrough status for this drug.
Right. Wonderful. And so does the drug work systemically, or is it somehow targeted to the liver?
It works pretty much systemically. We've shown it can benefit in various preclinical models, liver, lungs, kidneys, brain in stroke models, things like that. So GI lining. So it protects multiple organs.
So interesting. I love learning about. Yeah.
I think the biology is fascinating, right? We've learned about all these things our whole lives, right? And there's some literature out there that one can look at to understand a little bit more about the epigenome, and
right.
And this is a sulfated oxysterol. So there are even some pieces that have been written on dietary circumstances and people living longer and doing better. On just really to go on a side here, if you don't mind for a second, there was an article written on this where it talked about the Mediterranean diet and the diet in the Rift Valley in Africa and in Japan, and people living longer and doing better, right, cardiovascular disease wise. Well, this one scientist, a physician, she basically was attributing it more to less than the diet, but more to where these people were living, and these are all volcanic areas, and there's a lot more sulfur in the soils. And so she has speculated that sulfur is the key molecule here, and sulfur is certainly the key molecule. We think about phosphorus, think about a lot of different molecules being key, calcium for sure. Don't really think much about sulfur, right? And yet it certainly is in this circumstance. So it's really kind of fun to peel back the layers.
So interesting indeed. And so could this compound be applied in other indications, expanded to other indications?
I think so. We have shown all non-clinical now, but really impressive data with things like the acetaminophen toxicity, with an acute stroke, with acute pancreatitis, with acute kidney injury. In all these circumstances, we see pretty remarkable data. It's like lights on, lights off. If the drug's given, the results are tremendous. So it wasn't surprising that we saw these impressive reductions in mortality. We certainly hoped for it, but there aren't any good animal models for AH, right? This is more of a human circumstance. And so, but we know multiple organs are damaged, and we know all of the things that are going on. And so we had suspected that it would be a good place to test, and indeed it was.
Wonderful. And so I think you touched upon this and we talked a bit about this, but how have you really seen the field of epigenetics evolve from proof of concept in the lab and now to developing therapeutics that, are entering the clinic? How have you, since you've been in, with DURECT and developing these for a while, but I would just love to hear your perspective on just seeing the evolution.
We're just at the beginning. The way I think about it is there's a room with a locked door that we've been able to open now and just crack that thing about a quarter of an inch, and we can just peek inside the room and look inside there and say, wow, that's amazing. But there's so much more to be understood and done. If you think about this one molecule influencing 1,000 genes up and down regulation of them, right? And we're looking at a family of molecules behind this because of the insight we have into changing of DNA methylation. We look at the possibility in oncology and some other areas, but it's very early days. I mean, the biology is unbelievably complex. I heard a hepatologist speaking one time, a very well-respected physician, and the expression they were talking about methylation in the human hepatocytes, and they said there were 22 million sites of methylation in human hepatocytes.
Wow.
So you start thinking about the biology of that, right? And start putting, the numbers around influencing that. And sometimes talk about big data, right? I mean, it becomes really complex very quickly. So it's nice to have something that's already been there, that's out there biologically, that's highly conserved across all mammals we've tested, that seems to have this nice beneficial effect. That to be able to use that as our, that's our foothold, right? And from there we can. But it's just the beginning.
Just the beginning. Wow, I love that. Which makes it ever so much more exciting, for sure.
Oh, I'm unfortunately not at the beginning of my career, but what did I work? Because I think it's just an amazing place to spend a career is in this, and so it's the kind of thing I can, I'm naturally caffeinated, so I can stay up late. I could work, a lot on this for a long time.
That is so great to hear, and very exciting. And so what can we expect in the next 5 to 10 years, not just in the space of epigenetics, but from directly?
Well, I think for us, our focus, we have to be focused right now on larsucosterol and AH, because there's just such an unmet need.
Yeah.
We're talking about 30 to 40,000 people dying every year, average age 44. I'm constantly reminded in our first study we did, a Phase IIa, that was done down in San Diego, and there's a physician who treats these patients down there. He's a wonderful human being, and he does everything he can for his patients. He described to me a patient that she was 26 years old, went on her bachelorette party, and came back with AH. And now instead of planning her wedding, she's got a 30% chance she won't survive. And so, to be faced with this and no therapy out there. So it's incumbent upon, we have to do this. And that's what we're focusing on, getting the funding and getting that trial started. If we can do that, we should be able to finish that trial within 2 years. We have fast track designation by the FDA. So if the trial data are similar to what we saw, and what we saw was, almost 60% reduction in 2 different arms, right? 2 different doses. And so we looked at the odds, and what are the odds that that's just random, right? And so we asked our biostatistician to pull. We had 232 of the 307 patients were in the United States. So we said, if you pull out of 307 patients, 232 patients, do that randomly. We did that 10,000 times and looked and say, what are the odds that you're going to get these kind of data, right? And it only was randomly achieved 17 out of 10,000. So that tells you pretty good. That's a
pretty good odds.
Signal.
Yeah, signal there.
So that's what we're doing. We're pursuing AH for that, for AH, and then we'll hopefully have data at the end of 2026, submit the NDA, and get that on the market. From there, we obviously can start saving lives and change that circumstance, but then expand that out to these other acute uses in things like potentially acute kidney injury or stroke or sepsis or other things I was talking about, acute pancreatitis, for which there are really no good therapies today. And then medical therapies anyway. And then from there, use the insight into the epigenome to expand beyond that. But, right now we just have to take care of what's in front of us. If we don't, you don't get the next step. So the first step's the most important, and that's where.
Indeed. And I think you're just a few steps in after the first step. So that's really exciting. Yeah. And this is kind of just ending off more on a kind of a personal advice kind of a question, but what advice can you give people entering the field, and either in academic research or in industry? And I know there's a lot of transitioning happening now between academia and industry as well. So if someone is looking, a burgeoning researcher looking to start up their own biotech or pharma company, what advice would you give them in today's landscape, especially?
I would give them almost the same advice, in fact, the same advice that I was given 30 some odd years ago. And that is really, it's all about the people. With real estate, it's location, 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 right? And it's the same for starting a company, a biotech company. It's about the people, and the people, because you can find technology that it's around, and money comes and goes depending on the market cycles and everything else. But if you've got the desire and you've got a good asset, you'll eventually figure out a way to fund it. But you need the team, and you need men and women that you can trust, that you enjoy working with, that you want to work with, that you can be in a difficult circumstance with and come out and you can laugh about it and make it work and figure out a way around it, right? You don't want to be people who are going to be at each other's, bothering each other at each other's throats and the like. I'm so fortunate at our company. We have a number of people who have been with us this entire time, 25 years plus. A number of people I've worked with for more than 30 years. And so they're some of the best people I've ever known at whatever they did. My head of operations, she's just amazing. I've had great fortune to be with a lot of really good people over time. And so if you can gather those people together and establish a camaraderie, that's really what that's the most important thing you can do. And it's a way for life, right?
Yeah.
I mean, we only get to go through once, so you might as well enjoy it,
Exactly.
right?
And surround yourself with people, like-minded people, and have those support networks and really be able to work together. And I think this is also a testament to your leadership here as well, that you have people at your company that have been with you since almost day one. That is very telling. Yeah. Absolutely.
Yeah, and we've been through some difficult times, as you can imagine, right?
Absolutely.
There's downturns and all the rest of the turns that can occur.
And I think you've been holding it down very well at DURECT. So, Jim, thank you so much for your time. And it was so wonderful learning from you. And we look forward to staying in touch and learning more about the updates from DURECT.
Thank you, Ayesha. And anytime, I look forward to catching up. So thank you so much for an opportunity like this.
Sounds good. Thank you once again. All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.